right, so we're going to get started. My name is Lauren Stiles. I am the founder and president of Dysautonomia International, and I'm your moderator today. And we are really, really excited to have you join us, our guest expert speakers, as well as all of our audience members, to talk about some groundbreaking new POTS research that was done by Dr. Johansson and Dr. Fedorowski in Sweden, which is where they are tuning in from. So it's kind of late at night for them, and we appreciate them taking their dinner time to educate us. I just wanted to mention that this research that we're about to hear about was able to be funded because of all the generous donors that support Dysautonomia International. And so this is your research that you guys funded as a patient community. And we're just excited to be able to do that. And before I turn it over to our experts to present, I did want to plug that it's the holiday season, and we're having our holiday year-end holiday matching grant challenge right now. So to help us fund even more great POTS and other types of dysautonomia research in 2023, we are encouraging everyone to give what they can at curedys.org. curedys.org. And all of the donations between now and December 31st will be doubled thanks to generous matching grant donors, the Menzen family, and another anonymous donor who we have decided to call our Secret Santa double matching donor. So if you can, get over there later at the end of the session and try to support more research like this, because Dr. Johansson and Dr. Fedorowski and many other great researchers have a lot more important work to do to help us figure out POTS and other types of dysautonomia, and we want to be able to continue supporting their good research. So with that, I'm going to turn it over. And so a brief introduction. I think their expertise is pretty well known in our community. If you've been diagnosed for a while, you've seen other research papers, especially from Dr. Fedorowski, but Dr. Johansson is a postdoctoral fellow and a MD PhD at Lund University, which she just taught me how to pronounce correctly because I've been calling it Lund in my New York accent for many years, but it's Lund, and in Sweden. And Dr. Fedorowski is affiliated with Lund, but also affiliated with the Karolinska Instituet. And he is one of the world's most well-known POTS and syncope researchers. And we're so glad that they're interested in studying these diseases, because there are not too many folks in this space. So with that, I will turn it over to them, and I'll turn off my camera. Do one of you want to go first? I know we're going to talk about the proteomics study. That was pretty exciting, and also the new Malmo POTS score, which I think we'll get to a little later. Is that the order you want to go in? Okay, great. So I'm going to.
Sorry, Lauren. Perhaps is it okay if Artur starts with the maps, and I can just.
Sure. We have time for both. And also I wanted to mention to our audience members, if you have any questions during, so people have already started asking questions. So you can type questions into the Q&A at the bottom of your Zoom panel. And after the presentations, we will ask the researchers as many of the questions as we can get to in the time allotted. And unfortunately, there's hundreds of people on the webinar, so we probably won't get to everyone's question, but we ask that you keep your questions kind of generic. They can't give individualized medical advice, unfortunately, so please keep your questions general in nature. Okay, so I will pass it over, and Artur, I think you should be able to share your screen.
Okay, so actually, I haven't prepared any.
Oh, you don't have any slides. Okay, you just want to talk about the map.
I can share them later when Madeleine will start talking about the proteomic study, of course. So I can make some brief introduction. I can prepare some slides to illustrate what we are talking about. So to start with, it was quite amazing story. So a few months ago, or a few years ago, actually, when we started some case control studies, including POTS patients in Malmö, Sweden, at one point we realized that to quantify the symptoms, the symptom burden in this group was quite problematic, and this was due to the fact that there was no good symptom score on the market. So you may believe it or not, so we decided to find one. And what we found was one very sophisticated and wide spectrum score system for autonomic symptoms called COMPASS. And this was not really good for POTS only. And then we turned our attention to OHQ, which actually is designed for orthostatic hypotension patients. So then one point we realized, no, this is not good enough. We need something more, something more adapted to typical POTS patients. So what we did was a sort of a brainstorm. At that point, Victor Hammerfors and Jasmina Spahic, my PhD student, and Victor Hammerfors, my research partner, who now took over the Syncope Center in Malmö, actually. So we were sitting and discussing what we should do. And then we decided to look at the study which was published in the same time, around the same time, the phase of POTS, or the big POTS study, which I had a privilege to write an editorial for. So in this study, in almost 2,000 POTS patients, they reported on their symptoms. So we decided to extract 12 most pronounced or most prevalent symptoms in this group. And we decided to build a score. The score, quite usual score using visual analog scale from 1 to 10 points, and then to let patients decide where the symptom burden was situated on this scale in 12 main domains, like dizziness or lightheadedness on standing, and palpitations, representing orthostatic intolerance and heart rate increase, then fatigue, then shortness of breath, cognitive limitation or impairment. And then we ended up having 5 cardiovascular symptoms and 7 extra cardiovascular, non-cardiovascular symptoms, which was quite interesting. And we decided to test it on a group of 65 well-defined, well-diagnosed POTS patients. And as a comparator, we decided to include age and sex matched, completely healthy individuals. And the results were quite striking. If you look at these people, POTS patients and healthy controls, on the surface, they are almost the same. But if you look at the symptom burden, so the maximal score you can achieve in this symptom score is 120. So the average symptom burden was almost 80 for POTS patients and around 14 for healthy controls. A huge difference, a huge, really huge difference. I will show you a few diagrams from this study. So this was a good. We got a hit here, in my opinion. We caught the real essence of POTS symptoms, and the patients declared that the symptom score was very well adapted to the symptom burden, to the different symptom groups as well. What was even more interesting was that the average symptom burden in different domains was almost at the same level in all 12 domains. So not only lightheadedness and deconditioning, fatigue, but sleep disturbance, gastrointestinal problems, they were around 7, 8 on the 10-point scale in each and every domain for the whole group. So we realize that the POTS patients, they don't only have cardiovascular symptoms, but also other organ symptoms in the same and to the same degree. And the average healthy control, they almost don't have these symptoms. They have some symptoms, 1, 2 point on 10-point scale. And the separation was really good. If you put the threshold level at 40, so you will not see any healthy controls having a score or symptom burden more than 40. And the average is 14, of course. So if you let any patients to fill out the questionnaire, so you may be pretty sure that the average score or the minimal score will be well above 40. So we are, in our opinion, scientists and research groups in the world, they should try this out and start using it in the research so that we can make our populations comparable, so that we can compare different groups in different studies. And in this way, we can we can verify findings made in one location by other groups in another location, like in Calgary in Canada, or in San Diego, in other continents in Japan, knowing that we are talking about the same patient group having the same symptom burden. So I don't know if you have any question. So I would be very happy to answer any question that you may have on this issue.
Sure, there were lots of questions. Let me pull some from the list here. So here's a good one. Can you post a link to the tool that is used to measure dysautonomia? So I'm not sure if this person means MAPS itself or something else, but we can put, after I'm done chatting, we can put a link to the research article in the paper. And I think you included the tool as like an attachment to the journal article, right? It's like in the supplementary material.
Yes, this is translated into English. So there are both Swedish and English versions attached to the supplemental material to this article. And this is free. The access is free.
Yes, so we'll put that in the comments in a few minutes.
You can test yourself and see how many points you will get.
Yeah. Are there certain symptoms that score consistently across POTS patients, regardless of the cause of their POTS? For example, whether we think it's due to Sjogren's or EDS or something like that. Are there any sort of subtype clusters that you see in POTS in your MAPS scoring?
Not yet, not really. On average, when we summed up all the symptoms in this group of 65 patients, we noticed that the average was almost the same in every domain, in each and every of the 12 categories, being around between 7 and 8, meaning that if you sum up all these patients, they will end up having on average something, or some of them have something in each and every domain. Mm-hmm. So there are no specific patterns, but it might be studied, of course, if you have access to well categorized patients. And of course, one additional comment, probably we will see a little bit different pattern regarding post-COVID POTS, as these patients report a lot of additional issues, just like fever or a lot of dyspnea as well.
Yeah. One of the things I've noticed in these there are some papers starting to come out comparing pre-COVID POTS or what you might call non-COVID related POTS to long COVID POTS. And there are some differences, but I think one of the things we have to be mindful of is that most of the non-COVID POTS patients that are in these studies are people that have had POTS for a long time. And most of the long COVID POTS patients in studies are people who are relatively new onset relatively new onset POTS, at least within the past maybe year or 2 years, or now we're getting into 3 years. And so I wonder, because we don't have lots of research looking at the long-term changes and what POTS looks like at year 1, at year 2, at year 10, for some people who still have it at year 10. So I wonder if the differences we're seeing in the research are in part because they're just newer onset POTS, and newer onset POTS looks a little different, but also each triggering event, like different viruses or other infections, or whether your trigger is, a car accident or a concussion or something like that, that each of those might have a different fingerprint in what happens, POTS plus this other stuff, right? Like concussive patients will have POTS, maybe plus some actual brain injury and some more cognitive or more headache type things. So I think it's really interesting. And I would say we need more research, right? Like that's the answer to figuring that out, is taking this tool you've developed and then applying it to much larger groups of patients to see how, following them over time, to see if their scores get better or get worse or stay the same. So that was a great question. Here's a good question. Does this tool work for children? Can you use maps on children?
Oh, we decided to include patients who were older than 18 years for different reasons, just like ethical clearance that we had at our institution. So, but I think it should work for younger patients as well. I would say that, in my opinion, youngest POTS patients I've ever met, they were about 12, 13, 14 years. So I have never seen any POTS patients being younger than 12, but it might be a bias as well because I'm an adult cardiologist. So maybe I don't see these patients in the first place.
Yeah, I think POTS, what we've seen just talking to even the pediatric clinics, it's definitely less common in the prepubescent children. But we have, in our support groups and working with doctors who see large numbers of pediatric patients, we have definitely seen, 5 and 6 year olds who have something that looks like POTS. There's no official diagnostic criteria for children under age 12, but generally speaking, children, if they meet the 40 BPM heart rate criteria that's used for older, adolescents, if they meet that and they have all the symptoms, I think it's, sort of treated like it's POTS until they're old enough to officially diagnose it. But I want to hop over to Dr. Johansson's presentation if we can, because I know that is, I think MAPS is super important. We're so glad that you developed that. And I love it because it's a one-page tool. So what we're hoping is that doctors will start using this clinically, that they'll just put it in their patient. If it's a clinic that sees a lot of POTS patients, they'll just put it in their clinic forms that you fill out before you go for your follow-up visit. And then we'll start to get some data over time, of following people. So thank you so much for developing that tool. So Dr. Johansson, do you have slides to share?
Yes, I do. And I can share my screen.
Great. Okay, I'm going to turn off my camera and turn it over to you.
Thank you so much. So first off, a big thank you to Lauren and Dysautonomia International for being so kind and funding our research project that I conducted together with Artur and some colleagues. And essentially, we have recently published a new scientific article where we identified new disease pathways involved in POTS.
Before you hop to the next slide, I just wanted to tell you, we see your whole.
Oh, sorry, I forgot.
Presentation mode. Yeah, great.
I forgot. Oh, sorry. Hold on a second.
It's perfect. It's perfect. We can go now.
Okay. So the article was published in a journal called Scientific Reports, and it's open access, and you can download it and read the whole paper if you wish. And here you can see the title, which was Plasma Proteomic Profiling in POTS, Revealing New Disease Pathways. And this title can seem a bit difficult to understand. So I have tried to make this more information accessible to readers that do not have medical knowledge. So the background of our research project was that POTS is caused by different mechanisms and can pose substantial challenges for physicians, which strive to provide treatment options for patients with POTS. And there are very few studies which have identified biomarkers which can be measured in the blood and used to identify disease, both the disease activity, severity, and response to therapy in POTS. And therefore, a greater mechanistic understanding is necessary, and also detection of biomarkers that can help with this. So the aim of our study was to identify the footprint of POTS and compare it with healthy controls. And we analyzed 393 proteins in the blood. And we used a specific analytical tool called mass spectrometry to look for these proteins. And the study population was a case control study in 130 individuals. We had 65 patients diagnosed with POTS during a head-up tilt test. And these individuals had chronic symptoms of POTS lasting for at least half a year. And they were enrolled in the study between 2008 and the beginning of 2020. And then we also included 65 healthy individuals as controls. And these individuals were matched with POTS patients based on age and sex. So you can see that approximately 80% were women, and the mean age of our participants was about 30 years. And the age range was from 16 to 62 years of age. And the analysis of the protein pathways in POTS was conducted in blood samples that were analyzed with this mass spectrometry tool. And it's a specific analytical tool that you can use to identify chemical substances in different samples, for example, blood. And as I mentioned earlier, we had a total of 393 proteins that we found. And then we tried to understand their molecular pathways in POTS. And we observed, quite interestingly, that out of these proteins, 30 were significantly and differently expressed in POTS compared with these healthy individuals. And out of these 30, 19 proteins were upregulated and increased, and 11 proteins were decreased and downregulated in POTS. And the pathways that were involved were particularly associated with platelet aggregation, pro-inflammatory states that enhance inflammation, and also enhanced heart contractility and thickening of the heart muscle, and also increased skeletal muscle expression, as well as increased adrenergic activity. So the first protein pathway, which was involved in blood clots, we observed significant upregulation of these proteins here highlighted in bold, and these are involved in, for instance, the glycoprotein 1b is a protein which serves as a receptor for the von Willebrand factor, and it's involved and helps to prevent bleeding. And in POTS, we found that these proteins were increased. Another protein is filamine A, which also interacts with the glycoproteins, and it's also involved with the von Willebrand factor and promotes platelet activation and clotting. And here you can see some sophisticated names of different proteins: the tubulin beta-1, integrin-linked kinase, and beta-parvin. And all these were involved and are involved in blood clotting formation. And quite interestingly, we found that another protein called cartilage oligomeric matrix protein, abbreviated COMP, which is involved in also in this coagulation pathway and can cause bleeding and clotting as well. And it was downregulated in POTS. And let's see here if I can change my slide. The next protein pathway that we identified was that individuals with POTS showed increased expression of proteins linked to heart contractility and thickening of the heart muscle. And these proteins were particularly called integrin-linked kinase and beta-parvin. And another protein which was downregulated was thrombospondin 4. And some animal models have shown that when you have a deficiency in this protein, it can also increase the chances of heart muscle thickening. The following pathway that we identified was a pro-inflammatory pathway that promotes inflammation. And the proteins involved in this pathway were beta-2 macroglobulin. And previous research have shown that patients that have increased levels of this protein are also, these individuals have ongoing inflammation in their bodies. And as most of you are aware of, some patients that develop POTS do this after they have had a trigger, an autoimmune trigger, for instance, a viral infection or a bacterial infection. And quite interestingly, we found a protein called lumican, which interacts with these white blood cells and promotes an immune response. And under normal conditions, this protein restricts and prevents bacterial infections, viral infections, and helps with the inflammatory responses. However, in POTS, this lumican protein was downregulated, and we hypothesized that this may result in an impaired anti-inflammatory response in POTS. And therefore, POTS patients may perhaps not be able to inhibit this viral replication that occurs. And then we have yet another protein pathway, which in this case was involved with increased skeletal muscle expression of proteins. And here we have another long, sophisticated name of a protein, myosin light chain 1/3 skeletal muscle isoform, abbreviated MYL1. And this protein is expressed in the skeletal muscles, and it's required for the proper formation and maintenance of muscle fibers and function. And in POTS, we found that this protein was increased. And we hypothesized that it might perhaps also explain why some patients experience muscle fatigue and deconditioning in POTS. And then we have the final protein pathway, which we identified, by analyzing these proteins in the blood. And it is involved with in this adrenergic activity. So essentially we have yet another sophisticated protein, similar name to the previous one. However, this myosin protein is involved in regulating the vascular smooth muscles. The previous one was for skeletal muscles, and this is for smooth muscles that you have in your blood vessels, for example. And in POTS, we found that this particular protein was increased. And in POTS, it is well known that some patients have this increased sympathetic activation, which also causes an adrenergic activity and surge of norepinephrine levels in the blood. And that might be perhaps one underlying mechanism as to why we see this. And now this is my final slide and some conclusions and clinical perspectives. As I mentioned earlier, there are very few studies which have identified promising biomarkers in POTS. And the therapeutic possibilities are usually focused on just symptom relief. And this study that we just published, we identified some important new proteins which are associated with the increased platelet activity, promoting inflammatory states, and also increased adrenergic activity. And we hope that this understanding will also increase our mechanistic understanding of the POTS syndrome. And also future studies would have to focus on analyzing these proteins in more detail and perhaps more to identify perhaps more targeted therapeutic strategies for POTS. So this was just a brief explanation of this research findings that we published. And if you want to delve into more detail, you can read this article. So that's all. Thank you so much.
Thank you. Thank you very much. Sorry, I was in my typing mode. Let me put my computer up here. So there's tons of questions, and Dr. Fedorowski has been typing away, answering as many as he can, and I answered a few. And so I think, the biggest issue that when we shared this research with our patient community on social media, the biggest questions we got, the sort of most excitement, was around the finding of the reduced COMP protein. So I'm wondering if you could maybe talk a little bit more about that. That was my first thought as well. Not that the inflammation and coagulation stuff wasn't important, but that was sort of something, at least I kind of already assumed was in POTS. The other finding was really new to this, field. And I looked up COMP to see, what does this protein do, and it's in cartilage and it's in blood vessels and it plays a role in tendon strength and so much. So since we have a third of POTS patients have Ehlers-Danlos syndrome, hypermobile Ehlers-Danlos syndrome, and another subset, sort of undefined, loosely more than half, have hypermobility spectrum disorder, which is sort of like EDS without meeting the EDS criteria. You have some hypermobility. What role do you think that COMP could be an important thing we need to study further to understand that EDS POTS relationship? And I just would love to hear your thoughts on the relevance of that finding.
Thank you so much, Lauren, for posing this important question. And just as you mentioned, the spectrum between POTS and Ehlers-Danlos syndrome is quite similar. And as you already mentioned, some patients also exhibit both conditions. And as we also found in this study, it's quite, it's quite interesting and very novel. So these pathways and molecular mechanisms are still unknown to us. So we are just starting to perhaps understanding more about these conditions. And I agree that we should definitely focus on trying to elucidate this pathomechanism further. And as we wrote in the article regarding the COMP finding, we hypothesized that since it was downregulated in POTS, it can result in perhaps defective hemostasis. So controlling, when you bleed, the body needs to control the bleeding so you don't lose too much blood. So the hemostasis, and then the thrombosis as well. So still very novel findings that needs to be identified further, definitely.
Yeah, this type of, and I want to really explain this to our patient community listening in, this type of research is it's like the first step. Like we did, or you did a broad look at proteins in people with POTS, and you see what you find. And then you sort of start to think about how do these protein findings relate to this disease, what we see clinically. And then you do follow-up studies to see, you know, for example, I'm pitching you your next follow-up study, maybe that you should apply for DI funding for, you know, do patients with lower COMP have more severe hypermobility, you know, things like that, where we do like a deeper dive. We found a hint, and now we have to kind of run down that trail and see where that hint leads us. And there's a lot of trails that came out of this study. I also, let's take some questions here. I'm so tempted to ask you all my own questions, but I'll ask everyone else's because this definitely was an eye opener of a study of a lot of cool things came out of this. Are these proteins that you found abnormal in POTS, are they known to be associated with other diseases? And if so, are there any treatments that might up or down regulate the proteins to a more normal level?
That's a really good question. And these proteins are, you know, all individuals have these proteins in their body. They are measured in the blood and everyone have them. However, based on, here we had a POTS population and a control population that was assumingly healthy. And the only difference is basically the level that you have in these proteins. And we are in very early stages of understanding the mechanisms behind POTS. And for instance, this study, which we just published, hints towards that there is a both pro-inflammatory state in POTS, but also increased blood clotting tendency. So I cannot say right now that we should treat or perhaps prevent blood clotting by administering aspirin or something, but we should definitely perform more targeted studies in the future and try to see if perhaps some treatments are indicated in these conditions. What do you say, Artur?
Oh, I can only agree. This is a very early stage study and exploratory, so purely exploratory study. And we would like to thank Dysautonomia International for helping us perform it. So I would like to add one word more that I'm very happy and thankful that I had such a talented former PhD student, now postdoc, as Madeleine, who led the study, who made great use of funding to complete this study. So these findings are very preliminary, and that's why we are very cautious when we present conclusions. But what is very important now is to validate these studies in independent cohorts. What it means that if you study the specific group in one location, geographic location, one population, and then you recruit controls from the same population, then you would like to see pretty the same results in another population. So you would like to have the study performed in Calgary or in San Francisco or in Baltimore or New York. and different populations, the same methodology, and we would like ideally to see the same results. And all the results that are consistent, usually they confirm some important biological message to us. Just look at this point, go on in this direction, just look for the answer in this direction. So if these are platelets that are overactivated for some reason, there is something, some underlying biological process making them being activated. So look for it. So perform some additional testing. Look why they are activated, hyperactivated. And if you confirm these findings, then we may start talking, having good rationale about treatment. Maybe you should try out aspirin in these patients that demonstrate some impairment or dysfunction of platelets leading to blood clotting and so on. So this is the next stage. So first, validate these results in another independent cohort, and then move on to mechanistic, we call them mechanistic studies, to understand what's going on behind the scene.
Yeah, I think that's so important for patient and caregivers to know, like, whenever we find something new and exciting in a disease, whether it's POTS or another disease, it's really important that other researchers at another hospital system or another university can repeat that study to make sure that the findings are consistent. And if they're not, then we have to sort of maybe do a third study. Like, it's very challenging. And the reason why researchers do this is, there's always things that maybe people in Sweden just have higher levels of this marker, or the particular patients that were going to their university had more inflammation, because Artur is very known for the antibody research in POTS, so we have to make sure that there's replication. But in the meanwhile, I would say your findings of increased coagulation or blood clotting tendencies, this is consistent with a smaller study from Children's National Medical Center that came out a few years ago. They had pediatric POTS patients who had been using PICC lines or chest ports to get IV saline infusions. And it was a pretty small number of patients, I want to say like under 40 patients, but they looked at these children had a higher rate of blood clots than all of the other children in the Children's National Healthcare System that were getting chest port or PICC devices for other treatments, like for cancer, they were getting chemo or antibiotics or things like that. So their numbers were that children with POTS had an 11 times greater increase risk in deep vein thrombosis, which is, a serious blood clotting problem from PICC line or chest port use. So that's another reason that, most POTS experts are very, very reluctant to give our patients chest ports and PICC lines unless it's just absolutely the last ditch effort necessary. And so now that we have 2 studies hinting at increased risk of coagulation, which means, increased risk of blood clots, I would want our patient community to know that and to talk to their doctors about that. Because maybe that means if you are going to get a blood clot, a PICC line or a chest port, that your doctor should know about these 2 studies and think about, is this the right device to put in you? And if it is necessary, should there be consideration for preventing blood clots with aspirin or heparin or whatnot, or just maybe even just monitoring you much more closely for that. So I think it's very hard for you guys or any doctor to give clear treatment guidelines that apply to everyone because of one study. But I think it's a red flag that we need to be on the lookout for. And the inflammation findings also, we have other evidence of that in POTS, right? We have some work by Dr. Blair Grubb and William Gunning showing elevated inflammatory markers in POTS patients. And of course, the work that Dr. Fedorowski and the late Dr. David Kem and others have done on the various antibody findings in POTS. So I think that what you've found sort of is a second way of confirming some things we've already identified. And I think that is important, and it's going to lead, hopefully you do more follow-up research on this. And send in your application, we want to keep funding this type of research. Let's pop into more patient questions in the time we have left. So here's a good question we get asked a lot. Salt is a really important part of POTS treatment, but there is some research suggesting that salt is inflammatory. So is this something we worry about in POTS? Is salt inflammation a concern? I told you I wouldn't give you hard questions, but I lied.
If you have a salt in a high concentration, it's going to irritate your blood vessel wall, of course. But in this solution or concentration that is being given to the patients, it will absolutely not cause any irritation or any damage to your vessel wall. So there's no worry about it. And of course, once again, I would like to explain it. So it's dependent on concentration. This is 0.9% concentration. So this is a very low concentration in this saline that you are giving your patient.
Yeah. So another question is, were there COVID patients in the study? And this person is asking this question, I'm sure, because there has been a lot of research, very publicly discussed research, that long COVID patients tend to have evidence of inflammation and coagulation markers. And there's some discussion of people developing kind of hypermobility and joint instability type problems after COVID. So can you clarify whether there were COVID patients in this and what that relationship might be even, down the road?
Yes, and as I noticed that you already answered this question previously on your Facebook page, and all these patients that were enrolled in this study were recruited prior to the pandemic. So it was from the beginning of 2008 until the early beginning of 2020, but none of these patients had confirmed COVID.
So related to that, for those of you who are familiar with the microclots research going on in COVID, which if you're on Twitter, #TeamClots, there's a lot of cool, interesting research discussion happening. And Twitter is, there's all these problems with Twitter lately, but I think it's a great way for researchers from around the world to share info with each other and connect. And we've seen a lot of that. But that microclots research is largely being led by a doctor in South Africa, Dr. Restia Pretorius. And so very early when her first papers came out on long COVID patients having microclots, I reached out to her and said, hey, can we send you some non-COVID POTS blood samples so you can see if our, before COVID POTS patients might have this too. And she said, yes. So we have sent Dr. Grubb and Dr. Gunning. We're already studying platelet activation in POTS funded by Dysautonomia International. So they had already collected the types of samples she needed. So they have sent her some specimens, and I hope we will have a future, journal article coming out on that and a webinar to help explain it to everyone when it does. So I think that's there, there is a lot of research happening on different conditions that may be relevant and helpful to POTS. So that's kind of, good news for our patient community. And it would be really interesting to know if the markers you found are elevated in long COVID patients, right? So
yes.
Any chance you might be doing that at Karolinska or Lund?
Yeah, you are touching a point that I wanted to make. Actually, we are working on a cohort of quite consistent cohort of post-COVID POTS patients now at Karolinska. We have now collected around 300 patients or blood samples from patients diagnosed with post-COVID POTS, or definitely with POTS diagnosis post-COVID. And now we are working on recruitment of healthy controls that are age and sex matched so that we can use them as a comparator for this study. So we are definitely planning to perform mass spectrometry in the future on post-COVID POTS cohort, also as a sort of validation of Madeleine's findings. And if we find the same proteins upregulated, downregulated, then we will be very happy. Then we'll be very happy because then we may say that we have caught the signal that we should chase down to the bottom to find out the mechanism behind it. And I would like to make a little warning sign here. All these proteins, they may be involved in the disease. They may also be a sort of bystander. Yeah, what I mean is that if you're on a bus and you see an accident outside the bus, so your heart rate increases, of course, because you are affected by what you're seeing outside the bus. But you are not involved in the accident. You didn't cause the accident. You were not involved. But what's happening outside the bus affects you. And this is the same with biological systems. Something's happening outside the mainstream, and it may affect another biological process, that we then can catch through our sophisticated methods. And we see this protein is upregulated, and this is platelet protein. And then we may say, okay, so in POTS, the platelets are affected, but they might be affected by another process that we couldn't see through our methodology at this specific moment. So be patient. We will find out the way to catch the right signal in the end.
Yeah, it's very hard to be patient when we feel so sick.
I know,
and we know that you know that. We know that you know that.
I know that
but it is,
very well.
Everyone wants the answers
I see
yesterday.
patients every day, so I know they're very impatient.
So related to that, someone emailed me questions before the event. They wanted to ask, do we know if the autonomic nervous system plays a role in regulating these proteins? Could the autonomic nervous system itself, because we know in POTS that POTS patients have abnormal autonomic nervous system functioning. They usually have like too much sympathetic nervous system activity. Could that be playing a role in making these proteins abnormal levels?
Madeleine?
It's also a good question. Yes, it's a tricky question because right now we investigated these proteins just from one perspective. We had these blood samples and we performed this mass spectrometry analysis on them, and in particularly these proteins that were involved in a hyperadrenergic activation that we found. I think that these proteins, the myosin, one of these myosin proteins that I described earlier, perhaps could be involved. However, it's quite difficult to say anything beforehand because these mechanisms have not been identified and investigated before. So it's quite early and tricky, but work needs to be done to see if it is the case.
Yeah, bigger picture, not specific to these proteins, but bigger picture, there is lots of research historically on just general medical research showing that the autonomic nervous system does play a role in the coagulation cascade, and that generally speaking, sympathetic activation is associated with more coagulation. And if you think about it, I've given a slide on this a few times, people might have seen. If you think about it in a really evolutionary sense of things, like not specific to POTS, but just sort of human nature, why did your sympathetic nervous system have to exist, right? Because you had to run away from a lion or a bear, and you had to get your heart pumping and get your muscles all tensed up and ready to go. you might have been scratched or injured while you were fighting off that bear, and you needed to have coagulation to prevent you from bleeding out. So this is maybe, you know, my kind of, it sounds kind of cartoonish to think about it that way, but you know, there are really interesting sort of evolutionary reasons why the autonomic nervous system affects so many different systems in the body. So I'm hoping that that kind of future research you're going to do will dig us into that more. And similarly, there's a relationship between the autonomic nervous system and inflammation. That actually is a good question someone had asked in here. Is there anything patients can do now, even if we don't specifically know what's causing what, but there's this hint of inflammation? Is there anything patients can do, like at-home remedies that are pretty safe to try to see if it helps reduce their inflammation?
What do you say, Artur?
It's a very good question, but a very difficult one as well, because we had a lot of different hypothesis theories of muscle activation and people being on low histamine diet and people excluding gluten and other proteins, milk products and so on, and reporting that they feel better. Unfortunately, we have not performed any good study to confirm all these episodic observations. So I cannot give any recommendation, but what I used to do is that if a patient tells me, oh, I would like to test it, I would like to go on a low histamine diet or exclude milk products and gluten and different proteins and change my diet and see if it helps me, then I say, yes, go on, you do it and tell me if it helps you. If it helps, it's great. I'm very, very happy. And I think that there is some wisdom in our body, that our body can tell us in some instances, oh, we can hear some voice somewhere in our head, oh, please, if you can exclude it, you might feel a little bit better. So there might be some interconnections in your central nervous system telling you that this specific product is probably not so good for you. And then you may change your behavior and try to avoid it. And then suddenly you may feel better. So I would never diminish, I would never ignore such ideas. But the truth is that we don't have any good studies, controlled studies, to be frank. Yeah,
that's I would say that was a very doctor-like answer. You know, we don't have good research to confidently give recommendations to people to say, take this supplement, this reduces inflammation in POTS, right? We don't have that. And we want to have that. We have actually funded 2 studies on low-dose naltrexone, which is sort of popular amongst our patient community and other people who have sort of in conditions associated with inflammation and neuropathies and, you know, sort of in the world of POTS. So we funded 2 studies. One of them is with your colleague, oh my gosh, I'm blanking on his name, right? Marcus, and I can't remember his last name. What's his last name?
Marcus Stolberg.
Yes, thank you, Dr. Stolberg. Inappropriate for me to call him Marcus. Uh-oh, we lost. And then we funded a study at Dr. Raj's lab in Calgary. So there's 2 studies on low-dose naltrexone going on to see if that helps. The one at Calgary is a POTS-only study, and the one in Dr. Stolberg is doing is a low-dose naltrexone in long COVID POTS. So we'll get some, you know, evidence-based answer on whether that's helpful in POTS or not. And that has anti-inflammatory properties, right? And my thoughts as a patient is just, you know, things that we don't like to do that are hard are usually the anti-inflammatory things. Regular exercise is overall anti-inflammatory. Good sleep is anti-inflammatory. Eating a super healthy, nutritious, focused diet, no junk food is sort of anti-inflammatory. These are really hard to do, right? To sort of live the perfect healthy lifestyle, especially when you're already sick and you can barely make yourself dinner. Do you have a final closing thought for us as we come up on the time limit here?
Should I show this diagram that I promised in the very beginning? I don't want to leave anybody unsatisfied with my promise. So just a brief note. I wonder whether you can see it now on your screen. So this is the MAPS paper. As you can see here, there are two different figures here. These guys on the left side, they are healthy controls, and these guys on the right side are POTS patients. And you see that the overlap is minor here. And you see this group to the left, on your left-hand side, on your right-hand side, and healthy controls in the upper diagram, in the lower diagram, POTS patients. And you see they are different. Healthy controls, they are not at all affected to the same degree as POTS patients, but there are some outliers as well, people that probably don't feel well in general. And this is the major concern with this symptom score, is that when we looked upon post-COVID patients without POTS, so some of them may have very high score here, around 80, without having POTS. So this is more or less a system or evaluation system for very wide spectrum symptoms and high symptom burden in patients with underlying dysautonomia affecting cardiovascular system as well. But for the diagnosis of POTS, you have to perform non-form of orthostatic test, tilt testing or active standing test to confirm the diagnosis, and then you can put the symptom score on top of it to get the whole picture, meaning the cardiovascular disorder and the symptom burden. But this was all what I wanted to mention here.
Great. Thank you so much. To everyone who asked, there's hundreds and hundreds of questions that were asked, and we unfortunately can't get to all of them, but a lot of the things that I'm seeing that are asked are things that we have addressed in prior Dysautonomia International webinars. So I'd encourage you to visit our autonomic disorders video library, which is vimeo.com/dysautonomia, or find Dysautonomia International on YouTube. And we have lots of expert lectures like this addressing everything from like the basics of POTS for newly diagnosed or not yet diagnosed people, to lectures on Ehlers-Danlos syndrome and mast cell activation syndrome and autoimmunity. We have about 200 lectures on there. So there's a lot of, fall asleep at night listening to our webinars maybe. So I just wanted to thank you. Oh, she came back. Okay. I just wanted to thank you both for your time so much. Maybe we should ask her if she, Dr. Johansson.
Thank you. Sorry, my Wi-Fi just vanished.
Okay. Do you have any, Artur just gave us some closing thoughts. Do you have any final thoughts you wanted to share with our audience? Words of wisdom for the future?
Yes. First off and foremost, thank you, Lauren. Thank you, Dysautonomia International. Thank you every single person that has ever been able to provide some kind of funding for POTS research. That's greatly appreciated, and it means so much for us researchers to conduct this research because it's so valuable, and you make an incredible job, and thank you for existing, essentially. You're awesome.
I think the feeling is mutual. I'm going to speak on behalf of the 600 people or something that were on this call. So we appreciate that you guys exist too, and that you're doing such good work. And we hope we can continue to support it. Last plug for the fundraising. If anyone, we know everyone has different economic situations, so there's no pressure to give. But if you're in the position that you can do so, to contribute at curedys.org. That's Dysautonomia International's donation page, and you can specify that your donation is for the POTS Research Fund, the Dysautonomia Research Fund that supports research on other autonomic disorders besides POTS, and the Long COVID Research Fund, or if you're more interested in supporting our physician education outreach and our patient support work, there's an option for that too. So we encourage donors to tell us what they want us to spend their money on, all the donations are doubled until December 31st, up to $190,000, thanks to our two very generous donors. So I hope that we kick off 2023 with even more exciting research grants and progress for our community. It feels very slow when you're the patient going through it, but we are, making steps in the right direction to figure this out, to get us to closer, more targeted treatments that are more effective for these patients. So thank you, everybody. Have a wonderful holiday season, and we'll see you on the next webinar.
Thank you for listening. Thank you for your invitation, Lauren. It's great. And thank you for
so
being,
much.
Great.
being
Thank you
with
so much. It was great to have
our
you guys.
patients as well.
Thank you. And now you can go home and have dinner with your families. We've held you hostage long enough.
Thank you.
Thanks. Take care.